In this question, you have to identify the enzyme that is required to join a foreign DNA and a plasmid cut by the same restriction endonuclease to form a recombinant plasmid. Option A, polymerase 3. It is incorrect because it is involved in DNA replication. Option B, ligase. Now, DNA ligase is the enzyme that joins DNA fragments by forming phosphodiester bonds between the sugar phosphate backbones of the adjacent DNA strands. And this is essential for sealing the nicks between the foreign DNA and the plasmid, thus creating a recombinant plasmid. Hence, it is the correct option. Option C, EcoR1. It is incorrect because it is a restriction endonuclease that is used to cut the DNA at specific recognition sites. Option D, Taq polymerase. It is also incorrect because it is a thermostable DNA polymerase that is used in polymerase chain reaction to amplify DNA sequences, but it does not have the ability to join DNA fragments.